The Coral Holobiont response to climate change. Coral reefs make up less than 1% of the ocean floor, but are home to about 1/4 of all marine biodiversity. Providing sustenance to hundreds of millions of people worldwide, coral reefs also act as natural shock absorbers, protecting low-lying communities from wave surge. These magnificent ecosystems are, however, in danger due to human-caused climate change that is warming and acidifying the world's oceans. Prolonged heat stress can cause corals to eject algal symbionts, which provide them with energy. This process is known as coral bleaching because the corals turn a ghostly white color once the algal pigments are gone. Conserving reefs is therefore of paramount importance both to humans and to marine life in general. To help save corals, we first need to understand how they live, feed, and deal with short and longer-term stress. Like a human patient, in a coral hospital, we can assess coral health needs using a holistic approach that considers the individual parts as well as the entire organism, also referred to as the coral holobiont. The holobiont includes the cnidarian animal host, the dinoflagellate algal symbionts, the microbiome comprised of different bacteria and other eukaryotes such as fungi and viruses. This complex mixture of organisms, their genes, proteins, and metabolites, together control coral health and resilience under normal and stressful conditions. So how do we study coral health given their immense complexity? By using modern omics methods such as genomics, transcriptomics, proteomics, and metabolomics. Here is an example. The Hawaiian rice coral Montipora capitata is a stress-resistant coral that can withstand bleaching because it feeds on algae and bacteria when its own algal symbionts are gone. Can we assess rice coral health using omics methods even without knowing the history of the coral colony? It turns out that metabolites provide powerful tools for assessing stress levels in these organisms. Our lab found that small molecules called dipeptides, 2 amino acids bound together, accumulate over time as corals undergo heat stress. Therefore, a low level of the dipeptide arginine glutamine indicates a healthy coral, and inversely, a relatively high level of the dipeptide points to a stressed coral. Other chemicals such as the dipeptide arginine alanine and CDP choline also are markers of stress. Beyond metabolites, we can measure coral gene and protein expression to find markers of stress. Given these tools, it would seem straightforward to devise coral conservation strategies, but this is far from the truth. Reefs evolved in dynamic environments that are constantly changing, so corals have adapted extraordinarily well to daily and seasonal fluctuations in light, temperature, and pH. However, humans have far exceeded the normal stress load on corals due to uncontrolled coastal development, pollution, overfishing, physical degradation of reefs, and of course, climate change. Restoring holobiont health is highly challenging when the corals are already in dire straits. Therefore, it is no longer sufficient to take small steps forward to save reefs. Therefore, we need to harness all the tools available to us, ranging from omics and genetic methods to stricter conservation practices, to come up with novel and far-reaching approaches to restore and protect existing corals and ensure they survive into the future.